So with this, I want to get to the actual topic of the talk today, how to go about to do a sex experiment. So first, I'm sure you're excited to try to use sex for your experiments. So how can you actually assess if sex is a suitable method for your project, and will it actually give you any answers that you could use for your future work? Then the part will be about how to prepare for the experiment. So once you've gotten beam time or have access to a lab instrument, what do you have to watch out for when you're doing the experiment? And then the last section will be about what can you do while you're performing the experiments? What kind of troubleshooting can you be doing while you're doing the experiment? Dimitri was telling us about the theory of small angle X-ray scattering and how actually very simple the measurement itself is. All what you need is your sample that's in solution, and you hold it into the X-ray beam and you collect the scattering data of your pure sample, and you collect it of the buffer. And if you have these two scattering profiles, you can subtract them. And Al will be going into the details how to actually do this with computer programs to get the subtracted curve. But the sample itself is very easy. You just need your sample in solution and take it to the X-ray beam. So at our beamline, and as well as at the Diamond beamline and ESRF, we all have the same type of robot, but all the other beamlines and also the lab sources now have automated sample loading systems in place that does the step of putting your sample into the beamline for you. So with our sample changer, what you do is here we can put in PCR stripes or bigger epi tubes or 96 well plates. So our sample just goes into normal holders that you're aware of. You close the lid. There's a needle here. The plate gets positioned underneath the needle. The needle sucks in the sample. It goes through this tubing here. This is then the view you see from the camera that's looking onto the sample. The sample is positioned within the sex capillary, and then the shutters are open here. The X-rays come, hit the sample, and are scattered, and then the long tube and the detector that Clément was showing us in his talk, that's where the data is then collected. So basically what you need is to know how much sample you need to fill this capillary when you're talking about sample requirements, referring to the amounts that you need. So if you have enough sample, and if you're not fighting for every microliter, we normally suggest around 40 to 50 microliter per sample. We then load up to 30, 40 microliters, and that's normally sufficient to get you a good data set. If you're really limited, you can go down to 5 microliters per sample. In this case, we load the sample and we fix it. That means during this exposure, we're not moving the sample. But normally we prefer to move the sample during the exposure time, just to make sure that we can reduce the radiation damage. So you might remember Clément's pictures of those bubbles that were showing up during a too long exposure with the very intense beam. So to prevent radiation damage, we normally flush the sample through it. So if you have 40 to 50 microliters per sample would be the optimum to prepare. And of course, always more is better. To just be able to do some follow-up measurements or some repetitions if you're uneasy about something. But also with all of this, the best is always before you go and do an experiment is to contact your local contact or the person who will be in charge of you during the beam time. And all of us, and also at the other beam lines, everybody's really thankful if the users actually approach them in advance and to plan them properly so there's no surprises during the beam time. Because it would be a shame that if you once you have the beam time is to just not be prepared properly and have the wrong amounts with you. So that's the amount, the concentration. This is also a question that's come up during the last 2 talks in the chat, is how much do you actually need? And Dimitri was also telling us that it's also proportional to the number of particles. So the scattering intensity is proportional to how many particles that you have inside there, which kind of makes sense. If you have more particles, the scattering will be stronger, your intensity will be stronger, your signal to noise ratios will be better. So the better the higher the concentration, the better your signal. And this is just an example where we have 3 different concentrations. And if you're looking at here, the fuzziness, so the signal to noise ratio is a lot better for the very high concentration. You see that you can go very far out we're still getting a smooth-ish curve with not too much noise, not too much interference. And at the lower concentration, this is more wiggly. So this is a very small protein, but these are also pretty high estimates. So normally the rule of the thumb is if you have around a 50 kilodalton protein, if you go to 2 mg per mil, you should get pretty good data. And of course, the question also is, what do you want to see? If you just want to see the oligomeric state of your protein, the changes that are happening here in the very low S range of the scattering curve, then of course you also need that high concentrations, because you just need to get the information from this area of your curve. If you want to see small changes due to ligand binding or something, then most likely if these changes will reflect into a subtle change, maybe in the higher S range, and then you'll have to collect higher data. So the good thing is also, we'll be learning about that from Al2 next week, or at the end of this week, is how to use the program Crystal or other methods that are available to check your theoretical scattering curve and just get a feeling of what you actually want to see before coming to the beamline. So you do the theoretical calculation to see what would a theoretical Sachs curve look like, and that will also give you information how high of a the S range you'll need to be able to interpret the question that you're asking. Back to the concentration, the most important thing is that your sample is still happy. So you can't force your sample into a high concentration, just that you can check it off your list. But if your protein starts to aggregate and doesn't like that high concentration, then it's also not worth measuring at that concentration. So generally is try to get your concentration as high as possible, but keeping your protein in a happy state. And as I said before, if your molecular weight's around 50 kilodaltons and you have a 2 mg per mil concentration, then you're normally on the safe side that you'll get a good signal. So as we also mentioned many times, is that the signal, the intensity of the scattering is dependent on your molecular weight. So larger proteins here, so like apothioretin, the 400 kilodalton protein, will give you a very smooth curve out quite far, or covering a wide S range. And the smaller proteins show already the increase of noise at a lower S range. So if you look at these 3 proteins at the same concentration, you'll see that the higher concentration one gave the better signal. So the next question is, in what kind of buffer can we measure? And the theorists would say the ideal buffer would be water, just to keep the background as simple as possible, to make sure that your contrast, which is a very important part of the equation for the scattering, is as low as, so the contrast between protein and surrounding solution is as high as possible. And the more things that you add to your buffer, the higher the background gets and the less the contrast between protein and background. So try to keep the amount of additives as less as possible. But of course, here your protein also has to stay happy. And if you're measuring in pure water, you're going to risk the problems of having pH jumps during your measurements if you don't have any buffered system. If you don't have the salts, maybe you'll have unwanted interactions between your proteins. So the question is basically best to pose yourself is, what makes your sample happy? Can I reduce any of these additives that I'm adding to my sample? But otherwise, the advantage of Sachs is that you can basically measure in anything as long as your sample is in solution. So you don't have to like change into a PBS buffer or tris buffer or anything. You can just keep it in the buffer that you're normally working with, and that you also do your other parallel analysis with. So even though the Sachs measurement seems very straightforward and very simple, this question about sample quality is, of course, one of the difficult parts. And this is one of the main bottlenecks in preparing a good sample for Sachs experiments. So here I just did a theoretical calculating of the 2 proteins I've worked with in my life, or 2 extreme proteins I worked in my life. So this is the Foldon protein. It's 27 residues, a very small protein, and the proteasome, which is over 600 kilodaltons and a huge protein complex. Now I just did a theoretical calculation. If I was working on Foldon and accidentally got some proteasome contamination in my sample, this is how it would affect the Sachs scattering curve. So in blue would be the pure Foldon curve, and if I add percentages of Foldon, because remember, it's it's dependent on the size. So the bigger the contamination, the more influence it will have on your overall scattering curve. So this is just to be aware. And the problem that many contaminations that we have is the oligomerization states or the aggregation, and this really influences your scattering curve. And at higher percentages, it doesn't even only influence the small beginning part of your curve, but you also have influences all across the different S ranges. So sample quality is a big issue, and this is something that you really have to keep in mind when you're doing your SACS experiment. But basically, the take-home message of the planning an experiment and asking the question, is SACS a suitable method that I should be applying to my research? Then basically the answer is yes. If you have 100 to 500 micrograms of protein, you'll be able to already be able to answer quite a bit of questions, and you can actually gain good information that will help you understand the other data that you collect on your samples. So if you now decide, yes, I want to do SACS, you apply for beam time, you're granted beam time, and now it's time to prepare for the experiment and actually come to the beamline and work on your samples. So there's different modes of access. Normally our users prefer the on-site visits. So this is normally it's a 24-hour shift. This is just showing one of our favorite groups from Denmark that come super prepared for their 48-hour shift. So this year is actually a timeline. All the people participating, they have when are they at the beam time, when are they preparing samples, when are they sleeping, and they have it all stretched out, and they really plan for all the details. They also make sure there's enough chocolates and Coke available at the beamline that they can go through these 48 hours. The other mode of access, which now because of the current situation will become more and more the normal way how to access the beamline, is through mail-in operation. And here you have other problems that you have to consider. You have to make sure your samples come on time. You have to figure out customs, and you also have to be able make sure that the local contact who will be placing your samples into the sample changer for you will be able to understand what are the samples, what are the buffers, how do they belong together. So this is a very good example how samples are sent to us, where we have nice color coding to be able to understand what's buffer, what sample, and how does this work. And the way to go about to preparing the best samples, it also depends, of course, again, what's on your sample. So in an ideal case, your sample is very stable at very high concentrations. So in here, the ideal way to prepare your samples would be to concentrate your sample to the suitable concentration, perform a dialysis, keep the dialyzed buffer and your sample, then freeze them to the beamline. At the beamline, we can offer to quick filter them or do a centrifugation step to remove aggregates that might have occurred or been formed during the shipping and storage state. We can quickly recheck the concentration, for example, using a nanodrop and measuring UV observation, and then make a dilution series. So we take the high concentrated, dilute the sample down, measure with the corresponding buffer that was sent here along from the dialysis step, and then do a set of experiments. So this is the preferred way if your samples are at a high concentration, stable at a high concentration. If you know your sample doesn't really like to be stored at high concentrations, then the way about would be to take the dilute sample, perform the dialysis, again, keep the dialyzed buffer, surrounding buffer, and then your sample at the low dilution, filter, store it, ship it. When it comes to the beamline, we then filter the dilute sample and then stepwise do a concentration. And while concentrating the sample, always take out an aliquot and put it into our measuring tubes until we reach a high concentration. Of course, in between, check the UV at these different steps that we have the concentration, and then measure at the beamline with the corresponding buffer. How to know if your sample likes to be in a stable state, or what methods are available to check for the quality of the sample. So the most cheapest and simplest way, of course, would be a Naiders gel. Remember, SES gels, we'd be dissociating the bigger parts. And these are really the things that we want to check for when we're doing SACS experiments, to make sure we don't have any oligomeric states or high large contaminations that would really contribute to the SACS scattering curve. So if you can do a native gel electrophoresis, or passing your sample through a size exclusion column, where we have the separation, where the bigger stuff come off the column first, and then the smaller parts. And there too, you can see if you have any dimers or higher oligomers inside your sample. Of course, size occlusion works very well if we couple it to static light scattering, because sometimes these bigger things might be low in concentration, but they might be very large in size. And this is something that static light scattering can really pick up if you have that attached to your size occlusion setup. Dynamic light scattering also works well to check for your sample quality, especially if you're trying out different ways how to store your sample, trying different freezing or thawing techniques, adding dynamic light scattering to just do a quick check to see what's happening. You can identify if you're increasing or decreasing the amount of disturbing higher oligomeric species inside your sample. And then a little bit more advanced and not always accessible, but maybe you have, there's more and more laboratories that offer it as a service. You can send in your samples for analytical ultracentrifugation to see what kind of species are inside your sample. And you can also do native mass spec, for example, to figure out what's actually going on inside your sample. So the bottom row is the more accessible ones for normal laboratories. These other two might be a little bit more advanced. And you have to just look around and see what other options there are in your facility to characterize your sample. So before, when I was talking about how to prepare the sample, I was also mentioning the buffer, and that that you would dialyze your sample to get this. And that's really the best method of choice, is to perform a dialysis to make sure that you really have a good background that you can measure and use for the background subtraction. And this is a really important step during your SUCS experiment, is to make sure that your buffer really does look like the buffer that your sample's in, to make sure that you can subtract any scattering that's coming from the samples. And this is also, I think, that surprises many people, how sensitive the method is, using the wrong types of buffer, and that just the quick remake the buffer often doesn't really work, because you won't have exactly the same amount of salt concentration that your sample was in before, and that the buffers really don't match too well. So dialysis is a good way to do it. There's, of course, stuff that you have to consider when doing your dialysis, stuff you have to take into account: the temperature at which you're dialyzing, how much time you'll need to do an exchange during the dialysis, the concentration, the volume, so also the surface area from your membrane regarding to the other side. And this is maybe something that's trying and really optimizing to see how you can perform your dialysis. To all these parameters should be considered while doing it. I won't go through all of them. They'll be in the slides that you'll have available and that you can look at. The ways to dialyze. So the most common, of course, is to use the tube dialysis, but for this you need larger volumes. But if you don't have larger volumes, you only have smaller volumes. There's the cassettes and the commercially available items that you can buy to do the dialysis, but these are sometimes rather expensive. What we often do, if it's really just a last step dialysis where you don't really have to exchange like big amounts of glycerol, or you don't have any big sugars or anything, you're just really trying to get the matching buffer for your final step, we just use a normal Eppendorf reaction tube, cut off the bottom, place our sample inside the lid, put a little bit of dialysis tubing across the lid and close it, and throw this into a beaker and use that for a dialysis. And that works pretty well. And also the harvesting is really simple, because you just turn the thing upside down, take a tissue to remove the excess of liquid on top of the membrane, and then you just peeks it with your pipette, and you can recover your sample without much loss. So this is a really cheap alternative to get a good dialysis performed. You can also just throw the cup into a 50 ml Falcon tube, for example, and an easy way to transport or prepare it the day before your experiment. Dialysis isn't always possible, because, for example, if you have something super expensive and you won't be able to add that into the dialysate in the big amounts, In that case, Viking is a very good alternative. So here you're taking the expensive ligand, for example, you're adding it to your protein sample, but you have to remember to also add it to your buffer. And here the tip is really to try to use the same pipettes and the same volumes to really get this spiking as accurate as possible in comparison to what you're doing to the protein. One option that sometimes works really well, sometimes doesn't go too well, is to use a concentrator. So you do a deaffiltration step. So when you normally use the concentrator, your flow throughs that's coming through will be actually matching to the flow through, to the buffer that your sample's in here. Sometimes, depending on your sugar or glycerol, this might not be so evenly distributed between the volume or the buffer that's being remained in the concentrator and the one from the flow through. But for some examples, it's worked very well when you haven't had any other much choice to get to the buffer. Then, of course, there's also the possibility to do desalting columns. So here there's some advantages in getting smaller and smaller columns. And that actually I've noticed in the past it's worked better. It's getting better. And you can actually also work with, so you equilibrate the desalting column with your running buffer and you pass your sample through, and then that buffer will actually match pretty well to the buffer that you use to equilibrate the column. You can also use your mobile phase from the size occlusion column as the running buffer. But here too, we can see that during the separation, also sometimes stuff happens to the different components of your buffer, and that this also doesn't exactly have to be the perfect match. But these are just alternative methods that you have to be able to prepare a good buffer while you're measuring it. But if possible, dialysis is a good way. Regarding the sample quality, so over the recent years, there's been more and more efforts and more people have now switched to doing SEC-SACS. And from the questions that we've had during the last two lectures, it seems also to be something that many of you are interested in. So it's size occlusion chromatography coupled to SACS. And yes, this is for many samples a very good solution, because during the SACS experiment, you're separating your column, your sample over the column. So you're separating the big stuff from the small stuff from the wrong buffer. And while this they're being separated on the column, they pass through the SACS cell and you can collect the data from it. So you're removing aggregates on site and collecting data from freshly purified sample. So what we do in Hamburg, and also many of the American synchrotrons now also offer this option, is that after the column, you actually split the element and you send some of that through the flow cell and the other part through an array of detectors that have the light scattering detector and a refractive index, differential refractometer, that just also gives you additional information. So with the light scattering information, you can get an idea of the molar per weight estimate, and with the refractive index, you can get another concentration estimate for your sample. And by doing the splitting, you're preventing the broadening of the peak. So if you'd first go through the column, all these detector arrays, and then to the flow cell, you would actually be rediluting your sample and maybe also risking that your samples, the species that you just nicely separated, will remix again. So the splitting worked out pretty well to show that you can collect this information in parallel while you're collecting the SACS data. Samples for this works very well is, for example, when you're working with membrane proteins and you want to remove the free micelles. If you want to remove aggregates from your monodispersed samples, this is very good, or also just oligomers. And if you're looking at complexes and want to remove subunits that aren't completely bound, you can, this is where SEC-SACS really comes in well, and it's an option that you should be considering when you're planning your experiment. Here too, the best is always to reach out to the beamline, to your local contact, see what kind of columns are available, see what is needed to use the system before you just show up. And something that needs a little bit more preparation than just coming up with your batch samples. So yes, SEC-SACS is an alternative strategy to study moderately polydispersed samples. The requirements for your sample are pretty similar to what you need for batch experiments. You'll have to remember to bring enough buffer to the beamline to be able to run it. And you should really optimize your SEC run at home to make sure that you're using the best column and the best separation strategy at the beamline, just not to, it's something that you can't really just do trying at the beamline. You should really optimize this at home and then perform the experiments while you're at the beamline with the optimized conditions. If you have enough sample, it's always advisable to also collect some batch data as well with your second just to have this in comparison and to see what then actually happened during the second run, what you could separate. Because with the second run, we also often risk that we have radiation damage when the sample's going constantly through the capillary and being exposed with the X-rays. We do see that we often run into radiation damage. So if your contrast can afford it, it's always best to add some glycerol or other scavengers to your running buffer when you're performing a SEXSAX experiment. So, because also when you're doing the SEXSAX, you should remember that this is just an analytical step. It's not a preparative step. So you can't come with crazy amount of a mixture and think that SEXSAX will be the golden solution. You really have to try to have it optimized in advance before you come and do the measurement. Radiation damage can be an issue, so please make sure that you can also collect at least of a diluted sample, some batch data, and also remember that not all samples are stable over the separation steps. So you can see that some low affinity complexes actually dissociate on the column, and that the complex that you want to study isn't there anymore because it dissociated while it was coming off the column. Okay, so now we've planned the experiment, we've prepared the experiment, and now we're coming to the beamline, or we're performing the experiment, and what are stuff that we can pay attention to and look out for. So in this section, I've prepared 5 test cases. I just want to walk you through showing you how the data was collected and seeing which troubleshooting was needed to get something out of these experiments. Just a small disclaimer here that this is all work of fiction. So if it has any resemblance to real users, this is all coincidental. So our first cases, we have the very relaxed scientist. He's studying calmodulin. And for the paper, he just has to verify that the crystal structure that he has, which shows that it's in the monomeric state, is the true state of the protein also in solution. So he comes to the beamline and wants to measure his sample and show that it's a monomer. He collects the data, gets a pretty curve, even though it's a very small protein, has a very nice Guinier region. So in the Guinier analysis, we want that this is a linear relationship, and we see that it is. So the relaxed scientist is happy and goes home and starts doing the calculations, but then gets a shock because from the I0, from the forward scatterings, we can calculate the molecular weight. And instead of the 16.8 that he was expecting for the monomeric, this is 22 kilodaltons. So it's not really clear if this is really pure monomeric or maybe a monomeric dimeric mixture if you're just looking at these values. And the problem here is that if you're using the I0 value to calculate your molecular weight, you really have to understand your concentration. You have to have a real grips of the concentration that you're measuring. And here's the sequence of calmodulin. And if you take a closer look, you'll realize that there's no tryptophanes inside it. So using calmodulin as an example to collect the UV, or to determine the concentration of calmodulin using UV absorption is a very bad idea, because since there's no tryptophanes, even PROT parameter, which we normally use to calculate the theoretical extinction coefficients, will even tell you that there's highly going to be an error because of the missing tryptophanes. So measuring the I0 molecular weight estimate is a very good way to understand if your sample is as pure as expected. But for this, you really have to be able to know your concentration. So in this case, our user can get more excited if he chooses a different type of way to study the concentration, or to have different concentration assay at hand. I won't be going through all these options, but I'll just have them in the slides for you to check up on if you're not sure about the way how you determine your concentration. At the beamline, we do offer the differential reflectometer that we can also use for those kind of proteins that don't have any tryptophanes, or also if you're studying complexes and you're not quite sure about the molar ratio and what's going on, using refractometer is a good way to check it. So when you're looking at the concentration, there's stuff that you should be considering. You should really have an accurate way to measure your sample. So you have to consider your sample composition. So do you have tryptophanes or not? Also the buffer that your sample's in. Does your additives change the absorption at 280, for example? And there's other stuff to take into account when figuring out. So here's the reference to a protocol that can help you choose a suitable method how to determine your concentration. So luckily, there's also different ways from the SACS data to interpret the molecular weight or to get molecular weight estimates, for example, using the Parrot method. And that's something that the scientist wasn't able to do to understand. Yes, so if he uses the Parrot volume, he gets a 17 kilodalton, which is very close to his expected 16.8 kilodalton. So he was happy at the end, he could show that he did have the monomeric state. So that brings us to the next scientist, the lazy scientist. Here the question was trying to understand the effect of adding salt to the measurement. So we're measuring lysosine in 30 and 90 millimolar sodium chloride. And the idea was, is there any changes that you can see? And yes, if you measure in 30, you get a RG from 1.0. If you measure in 90, you have 1.2. So yes, there is a effect of adding salt to your solution. So the scientist was happy with these two measurements, went home, and first got the shock when he was trying to interpret the data, because according to the expected RG, we're actually expecting an RG of 1.4 nanometers. So none of these two were actually in that range, and the interpretation of the data that got more confusing. And the thing, and what we have to remember here is that we often talk about aggregation, but we also have the opposite. So we have repulsion of proteins, where proteins come too close to each other and they repulse each other. And this is often seen in a downward curve at the SACS data. If we look at the low S, we see that we have, so the line with linearity, we see that we're going, have the downward trend. And therefore, it's really important to measure over a concentration series of your samples. So here at 11 mg per mil, we have a stronger downward trend than we do at the 2.75. And if you look here, you can see that it's going closer and closer to linearity when we're diluting the sample. So this is something that you should really check and take the time at the beamline. The measurement only takes 3 minutes. So doing additional dilution steps normally doesn't cost too much of your beam time, and it's really worth the effort to see what's happening on, and really also expected that you measure over a wider range of concentrations so you can explain what's going on. So here, if you do the concentration series at 30 millimolars and 90 millimolars, you can see that actually they all have the 1.4 nanometers as the expected RG. So this user also becomes happy once he realizes that he has concentration effects, and you should really be measuring at different concentrations. So this brings us to our third scientist, the ambitious and the very hasty scientist. So he's pretty well prepared. He's trying to study this peptidase, that after the peptide binds, there's a large conformational change from an open to a closed state. And this is a very suitable type of measurements that you can do with SACS, because these big kind of changes will definitely be visible in SACS. And he also prepared well. So he realized, if I do the theoretical curves and calculate the Dmax, I'll see the large Dmax for the open state, shown here in green, and then the reduced Dmax for the closed state after the ligand has bound. And this would be a great way to screen many, many different ligands and many different pHs. So the scientists came to the beamline trying to get the most out of the beam time as possible, and measured many pHs and different kind of peptides, and was just going to detect the Dmax, but unfortunately they were all over the place and not really close to the ones that he was expecting after his calculations. The question was here then, what buffer did you use? And he just said, oh, I used this buffer, but for all these different mutants, for all these pH values, for all these ligands, just one buffer. He's like, oh yeah, it's always the same buffer. And that's where you realize where the difference comes from, same buffer. So yes, it's on paper, it's the same buffer, it's the same composition. But of course, the bottle that you used for your measurement won't be the same bottle that, or the same 100%, the same composition that your sample is actually in at the end. So these are one of the steps where it's really important to do this final dialysis, where you dialyze all the samples against the same buffer at the end, and then you can use the same buffer for all your measurements. And this here is just showing what happens if you're taking the wrong buffer. So the sample was measured in tris and glycine, but if you just only subtract the tris buffer and you have an under subtraction, this is what it looks like in your log file and in your practice plot. And if you do an over subtraction, so if you subtract glycerol instead of glycine, you can see that the curves really change and that stuff happens, but not only here at the far S, but also in your low S. And this is the part that you're using to calculate the Dmax. So yes, if you have the wrong buffer, you're going to get wrong values spitting out from the tables when you're doing the measurements. So this is really something to pay attention to at the beamline to see if your buffers are matching. Do they make sense? Is it what you're expecting? You might not always have time to perform a dialysis at the beamline if you realize that's the problem. Maybe if you're anyways running sec-sax measurements, you could then, from the most important one, do a sec, or you can try to use the gel filtration, the desalting columns, or also a concentrator to see if you can still get a better buffer per the sample that you're doing. So to quickly summarize our first three observations at the beamline, it's always good to check if you have anything, if the values are the ones that you're expecting. Is your I0 in the range, your MV estimate from the I0, is that in the range of what you're expecting? Because at the beamline, you can still remeasure the concentration, you can still check up on your sample and see what's going on there. Then remember to measure enough dilutions, look at your dilutions, see, does this make sense? Maybe you need to add some dilution steps just to really understand what's going on. Make sure you don't have any repulsion. Maybe you're removing aggregates while you're diluting. So do the concentration series. And also at the beamline, check to see if your buffer subtraction looks okay. And if you're getting a good feeling that your buffers match, because there's some stuff that you could maybe remeasure. You could, if you're, example, adding salt to your buffer, maybe you just want to do some extra buffer measurements where you're just measuring buffer at different salt concentrations to see if you can really hit a better matched buffer. Okay, so that brings us to the last two cases. So the next one is our super scientist. He's fully prepared. He's and done all the pre-analysis, and he's done all the theoretical calculations. And what he wants to check is the pH dependency of oligomerization state. So at the neutral pH, he has the tetramer, and then he wants to see if he changes the pH, if it can dissociate. He has the PDBs prepared of the tetramer, dimers, and monomers, and then brings it to the beamline to measure. And unfortunately, everything looks pretty bad and nothing looks like what he was expecting. And this is often the case. It's just that we have the unexpected problem. Somewhere along the storage, the transportation, something unforeseen happened to the sample and it starts to aggregate. Normally not much what you can do. There are some small little first aid kits or first aid steps that you could do at the beamline. So you could try centrifugating and to hope that the aggregates that formed will precipitate if you have a long enough of a centrifugation step, and then measure the supernatant with your protein. You can take a spin filter. So here we normally also have the 0.2 micron spin filters available at the beamline, where you just do a 1 minute centrifugation step and your pure sample that gets filtered out, and the aggregates hopefully remain at the top of the filter. And then of course, this would be an ideal case to do sec-sax and do a side exclusion step right before your sax measurement to remove the aggregates from the whole sample. So yeah, our super user then realized that he had some storage transportation alterations that had happened. And there's some stuff that you can check before you're sending your samples off or do the storage test. So if you have time, you can also check to see if your protein's at the best pH to keep it stable over time. What concentration works better? Is it better to keep it stable at higher concentrations or maybe lower concentrations? And as I was saying in the beginning, there's the two different strategies that you can then use if you want to freeze it at the higher concentration or at the lower concentration. Maybe if you add some salts, a little bit of glycerol, something you can do to protect it, make sure there's no proteases inside that are actually denaturing your protein or chopping it up over time. Then there's also some proteins like quick freezing, other ones don't like quick freezing. There's also, if you're thawing it, sometimes a quick thaw goes better, sometimes putting on ice. So these are all stuff that you can test and optimize at home. And not only will your SACS experiments gain from this kind of time and efforts that you've put into your samples, but also all your other following up or your other methods that you use, your assays, and of course, if you're doing crystallography or EM or something, can all profit from a more stable protein. So it's worth checking in to some of these. So if you at the Beamline see that you have some aggregation issues, there's, like I said, the stuff that you can do. You can do the centrifugate, try to precipitate your aggregates. You can filter. You can see if there's still time to set up for a 6-SACS run. Sometimes you can just do enough dilution steps, and then you're lucky and the aggregates also dilute out. And then at the lower concentrations, you can get a signal which is pretty pure. And I think Al will also be talking about this tomorrow on Thursday. So this brings us to our last scientist, the brilliant scientist. So he comes to the Beamline. He wants to do a dilution series of ribonuclease and wants to check his ab initio model, or he wants to get an ab initio model of ribonuclease. And looks at the pipeline and gets a shock because here it already points out only one frame was used. So as Clément was saying, we normally collect the 20 frames after each other and compare these to each other. And then we can see if any radiation-induced changes occur between these single frames. And then we just discard the frames that show radiation damage and take the frames that don't and average them and use those as our working files. But of course, here, if we only had one frame used from each experiment, we can't already say that within the first frame we didn't have any radiation damage. So here you can see from ribonuclease, we have 6 frames taken after each other, each of 50 milliseconds each. And you can see how quickly between the first and the second and the third frame, we have very big changes in our SACS curves. So normally you would expect within measurement that they would all align nicely on top of each other, but here we have these big changes. And as we know, the big stuff influences the large distances. We're in a reciprocal space here with our SACS curves. So here at the small s ranges, we're seeing the influence of very big stuff. But here within one frame to the next, we have a huge contribution from something large that is forming during the measurements. And these are the aggregates that are forming and are induced through the radiation damage. So for example, so here the dark blue, we have an RG of 1.7 nanometers, and within 4 frames, it's already increased to 6.4 nanometers. And of course, also our Dmaxes show the change. So we have something very large after those first 4 measurements within our sample. So what we've mentioned previously already is that to reduce this radiation damage, what we normally do in batch mode is that we're flowing the sample. So here's just the same example I just showed. This was measured in fixed load. So we load the amount into the capillary, turn on the X-rays and collect the frames one after each other. And that's where we see these strong changes. If we flow the sample while we're measuring, we're removing the radiated volume already and we're measuring fresh volume. So that's normally a good way how to get the curves how we want to have them. So there's also other stuff that you can do. You can add so-called scavengers that help you reduce the radiation damage. The scavengers have some advantages, some disadvantages, and it's really up to you to test for your sample which one's the best. And you can look through these, and there will be a reference to this paper we've done on radiation damage that can give you some ideas if you have a problem with that. So that brings us to the end and the summary. So when you're at the Beamline measure, make sure that you really have your concentrations measured properly and that the stuff that you're seeing getting spit out by the pipeline really makes sense overall. Then make sure you've measured enough concentrations that they explain the situation to you, that maybe you have to go further down into the more dilute ones, or maybe a higher concentration would help, and you can try to concentrate your sample at the Beamline and remeasure. Make sure your buffer is good. At the Beamline, you could still try to set up a dialysis if you have enough beam time or also try to do a sec run or use the concentrators or a desalting column to see if you can get a better buffer. Check if you have polydispersed samples, you can try to remove the aggregates, but also then just remember that we can also work with polydispersed data, and we'll be talking about this also in lecture 6, but you will also have to understand your sample. So sometimes you won't be able to remove the polydispersity because it's just the inner way how your sample works. For example, a monomer dimer equilibrium, you won't be able to remove the dimers because the system will just always automatically go back to dimers, but you just have to try to understand it. So try to collect as much information as possible to really understand what's going on in your sample. And then have the take radiation damage into account and look for this while you're collecting the data to see if you can improve the collection strategy to reduce the radiation damage. So don't be too relaxed when you're doing the measurements. It's a simple measurement. You push a button, the robot does everything for you, but really do pay attention. Don't be too lazy. Really go the extra step and do enough dilutions, do enough, make sure you have the right buffer, do the dialysis. Don't work too hasty, really take the time to look at the results, but also plan your experiments well in the beginning, and also be prepared for the unforeseen. So don't get stressed out at the beamline. If there is aggregates, just stay calm and remember there's stuff that you can do to try to the situation and get a bit more out of your data. And then just also remember that just like you, the beam is very brilliant. So be prepared that you might get some radiation damage and think in advance what scavengers would suit to your sample. But really the best thing is that you know your sample and that you understand what your sample is and what are the limits of your sample. What's the concentration you can reach? What additives can you remove, add to make your sample be happy? And really, really important is to really know your question. So we often have people that come at the beamline and they measure their sex measurement and they then just smile at us and say, so, we're like, well, what do you want to answer? And then they just say, they're not really prepared to understand what is the information they want to gain with the sex experiment. And if you really understand what's the question, do I want to see a conformational change between an open and a closed state? Do I want to see the oligomerization? If I know my question, I can interpret my data very quickly at the beamline and say if the qualities are sufficient and if this is the best that I can get out of the sample at my beamline. So with this, I'd like to say maybe one day I'll see you at P12. I'm not used to just seeing my laptop when I'm giving this talk, but and these days stuff is a little bit changed. So maybe I won't be seeing you at P12, but maybe we'll be seeing your sample at P12. And of course, the small angle X-ray community is a large community. So of course, there's not only our beamline, there's many amazing beamlines out there. There's lab sources that are getting better and better and better. So we're basically here just to advocate small angle X-ray scattering, and we're just happy that you do it. So choose the most convenient beamline for you or your most convenient setup. I'll be adding to the slides some references that you can check up on. There's a new one that just came out this year. Dimitri and me wrote a beginner's guide to small angle X-ray scattering that summarized pretty much some of what I said. And here, the highlighted in red is a book chapter Sai and me wrote showing the sample and buffer preparation. That many of the slides I showed actually figures that we use for this book chapter, and can explain a lot how to prepare your samples. And like I was welcomed as a first-time user at the beamline many years ago, we're also very welcome to first-time users and people who are eager to try out in our user lab and support you in a way we can to get your samples ready and into the beamline.